Can NGS drive clinical adoption of biomarkers? As Professor Banawat said, the dynamic nature of knowledge is reflected in human progress and technological achievements. You as experts know all about this. Genomics is a field that has seen huge progress and technical advancement in the last decade, with next-generation sequencing as an example that stands out.
Good morning, and thank you for joining this session on companion diagnostics. I was really looking forward to this session because it's very special to be able to present to you, the specialists in the field of genomics, our insights and to answer the question whether NGS can drive clinical adoption of biomarkers. My name is Inès Beumer. I'm a molecular biologist by training and experienced in biostatistics and bioinformatics. It is a predisposition to cancer in my family that drives my passion to engage in diagnostics and in translational research. Continuing on my PhD in oncology, I dedicated my scientific career to biomarker research and diagnostic implementation in general. This fits perfectly with the mission of GenomeScan, where I'm accountable for innovation, product development, marketing, and funding projects. We are a leading Dutch NGS service provider based in Leiden, the Netherlands. As Dutch diagnostic hub, we perform NGS diagnostic services, both germline and somatic, for the majority of the Dutch academic medical centers. For pharma and biotech, we are involved in biomarker and drug discovery and development, where we commit to large custom projects that are tailored to the specific needs of pharmaceutical companies like Janssen and UCB. It is our mission at GenomeScan to make a difference in healthcare by leveraging innovative NGS technologies for these two markets and the academic research world.
For the obvious reasons that are well known to you as experts, the ultimate goal of medicine is the identification of disease and subsequent treatment of patients based on the clinical, pathological, and individual disease characteristics. For this, we need different types of biomarkers. So, which type of biomarkers could help? Recently, my dad received an invitation to participate in the Dutch population screening program for colorectal cancer. This is one of the examples of early detection screening strategies. For screening, one can think of assessing disease susceptibility, like BRCA1 and 2 for breast cancer. An example for definitive diagnosis and general typing is the complete blood count of an individual that experiences a high fever. This is in order to check for a bacterial infection. Prognostic biomarkers assess disease outcome regardless of treatment, like TP53 mutations to assess the likelihood of death that is associated with CLL. Effects of treatment can be predicted with predictive biomarkers, or companion diagnostics, for which lack of ER and PR presence in breast cancer is a well-known example. In line with predictive biomarkers, pharmacogenomic markers can help foresee the likely response to a particular therapeutic agent to guide choices in dose schemes. And the last type of biomarker involves monitoring of disease recurrence and therapeutic response, like PSA for prostate cancer progression. Now we must ask ourselves, what makes a good biomarker? Everyone would prefer drawing blood over taking a fine needle biopsy. Therefore, easy obtainability and minimal invasiveness are key. Confidence in a biomarker is necessary to align for clinical adoptability. A biomarker needs to be easy, simple, and stable, or in one word, robust. At the same time, it needs to be verifiable as a good indicator of the condition or risk, the progress or treatment effect that it aims to measure. And finally, we need to maximize healthcare effects given the resources available, as expensive biomarkers do not make the cut. You all know about the observation that many biomarkers have been published since the early days of high-throughput research, but only a few of them have been clinically adopted. Although translational research is envisioned to tackle the major challenge of translating basic research discoveries into improved treatment of patients, The need for new biomarker strategies for clinical implementation remains existing. This is particularly evident in oncology, where treatment modalities of cancer patients are mainly based on morphological appearance and the anatomical extent of disease at diagnosis. It took long before the internationally renowned guidelines started acknowledging the benefit of disease-driven biomarker assays, in addition to clinical pathological risk assessment. We as scientific community, with you as the genomics experts, can drive to change this. The question is, what is needed to do that? My answer to this question is that there seems to be a need for awareness. Awareness of the requirements for clinical adaptability and the key decisions in a typical biomarker design process, especially for those in the world of academic research that aim at biomarker discovery. Everyone working to discover and develop biomarkers should be able to answer the question, which steps are necessary to make a biomarker clinically adaptable. In addition, we need to utilize and master the power of advanced techniques like next-generation sequencing to our advantage, and keep on questioning if we can improve in relation to existing biomarkers. So how can NGS drive clinical adoption? Today's presentation will answer both questions, with a summary of the roadmap to clinical adoption of biomarkers that features next-generation sequencing. Technical advancements of NGS strengthen its robustness. This graph shows the technical quality scores over time of the three main platforms that we currently use at GenomeScan. These graphs demonstrate stability and robustness over time that is consistently higher than the standard industry cutoff. With the introduction of new NGS machines, in this case showing the Illumina machines that we operated, the sequencing runtime decreased significantly, while data output increased with every new introduction. In 2011, a single run took 8 days and yielded 400 gigabytes. Since the introduction of the NovaSeq 6000 in our lab in 2018, a runtime of 1.5 days yields 6,000 gigabytes worth of data. In the early days of NGS, the $1,000 genome was seen as the target that could enable mainstream whole genome sequencing. And since we passed that point in 2018, this vision is starting to become a reality. Different stakeholders are working together to get whole genome sequencing available for reimbursed clinical use in the Netherlands. Now we know that NGS holds great promise for technical advancements that are needed to drive biomarker discovery and development. I would like to take you along the path of the 9 steps with a real-world success story in the making as an example. 9 key steps define the road of potential biomarkers, starting with identifying a clinical question to finally the clinical adoption of the developed assay. Steps are dedicated to assay development, regulatory requirements, reimbursement requirements, and finally the adoption of the assay into clinical practice. Discovery is the step with the most scientific recognition and the highly cited publications. Following steps to clinical implementation require a lot of investment in terms of time, in terms of money, and in terms of years or even decades of subsequent work. Let us take a closer look at those 9 steps with a companion diagnostic for Parkinson's disease as an example. Parkinson's disease is the second most common age-related neurodegenerative disorder after Alzheimer's disease. The most well-known symptoms are tremors, personality changes, and memory loss. Prevalence is age-dependent and ranges up to more than 19 people per 1,000 among those who are 80 years and older. The most common genetic factor known to date for Parkinson's disease are variants in the GBA1 gene. These genetic mutations affect approximately 10 to 15% of Parkinson's disease patients and is a validated risk factor that generally leads to an early age of disease onset and more rapid disease progression. Existing treatments are symptomatic in nature and do not modify the underlying disease progression. and some treatment approaches are contraindicated for patients with GBA1 mutations. Therapies targeting underlying pathogenesis could slow disease progression in this population. The new compound presents the potential of being disease-modifying therapy for a genetic subset of Parkinson's disease. Biomarker discovery and development first starts with defining the problem. The highest chance of clinical adoption of the research finding after assay translation is when it answers an actual clinical need. And second, with defining which patients have this clinical need and finding the balance with the available resources, biomaterials and data for representation of the target population. Which patients are included, which are excluded, and why. In our example, the clinical need is a companion diagnostic for the further development of the GCase activator LTI-291. The companion diagnostic is aimed to identify patients carrying a GBA1 gene mutation as target population that may benefit from activation of this enzymatic activity. Sequencing was performed in the framework of a large program for 3,402 people with Parkinson's disease living in the Netherlands. Variant frequencies were compared with an existing Dutch control cohort of 655 people. A discovery strategy deals with the trade-off whether to use an unbiased or knowledge-based approach. The choice of technical platform, for instance leveraging NGS comprehensiveness, and available retrospective and prospective datasets. This is also the time to think of IP protection for the biomarker, which is a blessing to protect the investment that is needed down the line, and a curse because it costs a lot of money and sets back the timelines of publishing your findings. Discovery of the companion diagnostic in our example was knowledge-based because the target was selected, and unbiased because the entire open reading frame was assessed and not focused on known variant locations. Findings were published. This was done last September in the peer-reviewed Journal of Movement Disorders. The technical difficulty was to unambiguously sequence the functional GBA1 gene and not the pseudogene. The exonic region of the pseudogene shares a 96% sequence homology with the coding region of the GBA1 gene. This technical challenge is why searching for variants in the entire open reading frame of the GBA1 gene was not done before. An experimental setup that combines long-range RT-PCR amplification for short-read sequencing using Illumina equipment, long-read sequencing with the PacBio sequel for phasing, and subset validation using whole exome sequencing was the solution that enabled us to sequence the entire open reading frame. The study identified 18 novel variants and showed high prevalence of a potential Dutch founder complex allele. This highlights the importance of sequencing the full GBA1 open reading frame. These results facilitated recruitment for a GBA1 clinical trial. Further biomarker development is done in parallel to this clinical trial and outside our knowledge. The next step in assay development in general would be the translation step. This step takes a biomarker from research complexity to assay simplicity with clear yes and no answers. It also takes the biomarker from batch or cohort-based normalization to a setting in which single samples can be processed. The assay is a result of balancing between classifier parsimony, or low complexity, and striving for accuracy and precision, with the risk of overfitting the data. In the end, the goal is to confirm the findings of the association and provide clinically meaningful quantification and cutoffs. The facilitation step starts with the choice of central lab versus point of care setting. If applicable, software should minimize command line coding and maximize automation and flexibility. Next in line is the assay lockdown of established methods, procedures, and the intended use. This is required before the next 5 steps in order to enable the results of subsequent experiments, clinical studies, and trials to be valid. Also, arrangements for a quality control system that covers assay maintenance and monitoring is key. And in the end, beta testing, or also known as feasibility studies, will show if all logistic arrangements are in place. Regulatory approval also depends on showing the analytical validity of the assay by establishing that the assay measures what it intends to measure. This involves the limit measures and repeatability, precision, and reproducibility assessment in order to establish that the assay is reliable and robust. Clinical validity essentially answers the question, how accurately, consistent, and reliably can the assay classify samples of the target population? This involves retrospective and prospective clinical validation of the locked assay that is required for regulatory approval. At Genome Scan, we have a quality assurance program in place to ensure consistency in our high-quality standards. The clinical utility step should demonstrate clinical benefit by answering to what extent testing improves health outcomes relative to the current standard. This is done in prospective randomized clinical trials. A test itself does not have inherent clinical utility. Rather, it is the intervention that is guided by the test results that influences clinical outcomes and determines clinical utility. Some tests at first do not seem to have clinical utility, but their use seems justified because of other benefits. Testing for Huntington's disease, for example, offers no options for effective clinical management, but can be of help in life planning and reproductive decisions. Cost-effectiveness studies are a world on its own. They compare the new assay with the current standard at cost versus benefit level, and they distinguish between direct costs, productivity costs, and intangibles. Finally, when the assay is regulatory approval, it can go to market. In order to facilitate clinical use, the assay needs clinical adoption, and clinical adoption requires clinical awareness, clinical acceptance, and engagement of key opinion leaders, and also engagement of patients and their advocacy groups for the necessary feedback. Inclusion in guidelines is necessary for reimbursement and in turn for clinical adoption, and also think of suitable and useful marketing strategies. Development of new drugs and companion diagnostics can go hand in hand. Basic research and translational research can be aligned. In preclinical studies, both drug and companion diagnostic can be evaluated alongside. Further experiments, clinical studies, and trials that are required for biomarker validation can follow the clinical trial series of the investigational new drug. And arrangements of regulatory approval can be done simultaneously, while also cost effectiveness is assessed. And this all to aim for market introduction for the medical prescription drug and companion diagnostic in one go. Now getting back to the Parkinson's example, Lysosomal Therapeutics has developed a potential new drug and companion diagnostic. Clinical trial phase 1 has been finalized, and Biotech, the new owner of the Parkinson research program operated by LTI, plans to advance the compound to phase 2 clinical testing in 2021, with all efforts hopefully resulting in a first disease-modifying drug for Parkinson's disease. At Genome Scan, we can assist in all phases of discovery and development of drugs and biomarkers. With expression analysis and genotyping, we can help you characterize your patient cohorts and provide data for target identification, discovery, and prototyping. Pathway analysis can confirm intended effects and identify drug side effects at molecular level. And microbiome screening can assist during clinical trials to test for the influence of the compound on the patient's microbiomes. And authentication of your cell lines at all steps can reassure that your model system does not change at molecular level and monitor production quality of the antibodies or protein compounds during production. I thought it would be nice to present Genome Scan to you in more depth using some numbers. The start of GenomeScan, formerly known as Service Access, was in 2002. We are ISO accredited to ensure our high-quality standards. Our team currently consists of 45 young and experienced professionals. Since 2015, we sequence all diagnostic samples for the clinical genetics departments of the EMC and LUMC. To strengthen the collaboration, they became shareholders of GenomeScan. To stay at the forefront of innovation, we participate in EU-funded projects since the beginning. With the costs of sequencing decreasing, more research groups and companies come to us to incorporate NGS into their work. Our lab facility measures 950 square meters, housing the latest equipment for short-read and long-read sequencing. And last year we produced 425 terabytes of data. 44 hours is the runtime for an S4 flow cell run on the NovaSeq that delivers 3 terabytes of data, and after which it currently takes around 1.5 hours to analyze 96 whole exome samples.
I promise to answer 2 questions. Which steps are necessary, and can NGS drive clinical adoption? As summarized, there are key decisions to take in every step of the typical biomarker design process that can influence eventual adoptability of the biomarker. With the technical advances of NGS, the technique is robust, affordable, and fast, and can be used in every step of the discovery and development of drugs and biomarkers. Both medical prescription drugs and companion diagnostics can benefit from co-development. As scientific community, you can drive development of biomarkers and their clinical implementation for personalized medicine. You are aware of the steps that could lead up to clinical adoptability and know that NGS is an advancing technology that can be leveraged to get there. I would like to invite you to contact us to discuss your project.